Hello everyone, this is Dr. Kalpana and you're watching my YouTube channel Pharmacy Lessons Made Easy. In our series which was started for application of computers in pharmacy, this is another video where the bioinformatic databases are being discussed. So this particular topic is very important from your exam point of view, not only for the multiple choice question based but also for the big question, because from this whole unit bioinformatic databases is very much important. So let's discuss them one by one. It is very well said by Kenneth R. Miller that our own genome carry the story of evolution written in the DNA. The language of molecular genetics and the narrative is unmistakable. So what would happen if database didn't exist? We have a goal in bioinformatics to turn data into information and information into science. So we shall be going to talk about bioinformatics and their databases. Let's have a quick look at these. So firstly, understand what bioinformatics is. It's a new scientific discipline. It deals with the application of computer, which has now become the heart of modern biological research. It involves discovery, development, and implementation of computational algorithm and software tools that facilitate an understanding of the biological processes with the main goal to serve the humankind. In today's era, bioinformatics is rapidly developing branch of biology. It drives knowledge and is highly interdisciplinary in nature. It uses the techniques and concepts from bioinformatics, statistics, and mathematics as well. So it provides not only the sequence analysis but also the structural bioinformatics and the simulation of molecular interactions like metabolic pathways and Gibbs network. So the main objectives for bioinformatics are: it organizes data in a way that allows researchers to access existing information well and to submit the new entries as they are produced. Second objective is it develops tools and resources which help in the analysis of data. Third, and the most important objective of bioinformatics is to exploit these computational tools so as to analyze the biological data, interpret the results in a biologically meaningful manner. So let's understand more detail about it. So we have used the term database. What are these databases? Databases are essential for bioinformatic research and application. There are many databases which exist which covers various information types like DNA, protein sequences, molecular structures, phenotype, and biodiversity. So these databases may contain empirical data which is obtained directly from the experiments, or it can have a predicted data which is obtained from analysis, and most commonly both. So they may be specific to a particular organism, pathway, or a molecule of interest. There are many types of bioinformatics we have studied so far. First database is ENTRES. In 1991, ENTRES was introduced in CD form. ENTRES is a molecular biology database system. It provides integrated access to nucleotide and protein sequence data, gene-centered and genomic mapping information. It's a unique feature of this system that it uses pre-computed similarity searches for each record so as to create links to the neighbors or related records in other entries databases. Another database is EMBL. It was created in 1974 and it is an intergovernmental organization funded by public research money from its member states. It contains primary nucleotide sequence database in Europe, the European Molecular Biology Lab, that is what is being abbreviated as EMBL. It is a molecular biology research institution which is supported by 27 member states, 2 prospect states, and 1 associate member state. EMBL runs an active science and society program which offers activities and events on current questions in life science research for the general public and the scientific community as well. Moving to the next databases. So this one is our third database that we are discussing. Its name is FlyBase. Drosophila melanogaster has been an experimental organism since the early 1900s and has since been placed at the forefront of many areas of research. FlyBase is an online bioinformatic database and the primary repository of genetic and molecular data for the insect family Drosophila. FlyBase originated from a variety of resources ranging from large-scale genome projects to the primary research literature. For the most extensively studied species and model organism, Drosophila melanogaster, a wide range of data are presented in different formats. Moving to the next database. The fourth database is FSSP, which stands for Families of Structurally Similar Proteins. It's a database of structurally superimposed proteins which are generated through distance matrix alignment algorithm. The database provides a rich source of information for the study of both divergent and convergent aspect of the evolution of protein folds and defines useful tests. This database is helpful for comparison of protein structures. The URL is being displayed here. Another, the fifth one, it is OMIM. It was founded by Victor A. Marcus in 1960. He was from John Hopkins University School of Medicine. It contains genetic information of human disease and is continuously updated catalog of human genes and genetic disorders. The online Mendelian inheritance in man, which is the full form of OMIM, is a comprehensive, authoritative, and timely knowledge base of the human genes and genetic disorders, which are compiled to support human genetic research. Here is its logo as well. Moving to the sixth one, it's PIR. It was founded by the National Medical Research Foundation, 1984, Washington, D.C. It is protein information resource which contain annotated protein structures. PIR is located at Georgetown University Medical Center. It is an integrated public bioinformatic resource to support the genomic and proteomic research and scientific studies. It contains protein sequence databases. You can see its logo also, and the URL is also being displayed on the screen. Another, the seventh one is PubMed. It was founded by National Center of Biotechnology Information US in 1996. It contains biomedical literature information. The PubMed is a free search engine accessing primarily in the Medline database of references and abstracts on life sciences and biomedical topics. It's a very popular tool. You can see its URL as well as the logo is being displayed on your screen right now. Another one is number 8, that is SCOP, SCOP. It was founded by Alexei G. Merzin in England in 1994. It provides information on the structural classification of protein. They are classified on the basis of three-dimensional structures. It is largely a manual classification of the protein structural domains which are based on similarity of their structure and amino acid sequences. A motivation for this classification is to determine the evolutionary relationship between proteins. The SCOP database is freely accessible on the internet. So you can see its URL being displayed here and just look at its very catchy logo as well. Another number 9 is Swiss Prot. It was founded by Amos Barrick in Switzerland in 1986. It is curated protein sequence database. It strives to provide high level of annotations, means the description of function of a protein, its domain structure, the post-translational modifications, variants, etc. A minimal level of redundancy and a high level of integration with other databases are its unique features. The Swiss protein sequence data bank consists of sequence entries. The sequence entries, they are composed of different line types, each with their own format. So this was all about SCOP and SwissProt. Moving to the 10th database, that is TAIR, which was founded by National Science Foundation in USA in 1999. It's a Arabidopsis information database. The Arabidopsis information resource, that's why its name is TAIR, is a community resource and online model organism database. It provides genetic and molecular biological data. Basically, the model plant which is used is Arabidopsis thylania, and this is also called mouse ear cress. This database is very usefully integrating information about Arabidopsis genome, genes, gene production, natural variants, mutant alleles, and plant phenotypes in research literature. The data which is present in TAIR can be retrieved using simple and advanced research, bulk query, and there are certain download tools also which are available, and it is utilized in collection of prepared text files as well. You just look at its logo as well. It is indicating the plant that we are talking about, Arabidopsis thaliana, right? So that is being displayed on its logo as well. Moving to the next, that is the 11th number, which is Procite. It was found by Ms. Barrick in Switzerland in 1988, and it provides information on protein families and their domains. Procite's use include identifying the all possible functions of newly discovered proteins and analysis of known proteins for previously undetermined activity. See, the properties from well-studied genes, they can be propagated to biologically related organisms, and for different or poorly known genes, the biochemical functions can be predicted from similarities. The ProCite tools offer for protein sequence analysis. You can see its URL here as well as the logo being displayed on the screen right now. Yes, on number 12 is KEGG. See the more exciting logo for KEGG. It was actually found by Minoru Kenichiya in Japan in 1995. The Kyoto Encyclopedia of Genes and Genome is a database with related computerized information on biomolecules and cell biology. So this Kyoto Encyclopedia of Genes and Genome is a resource for understanding high level of functions and utilities for the biological system, like cells, the organism, the ecosystem, for genome and molecular level information. So that varies from genomic level to molecular level information. So it is providing all kind of utilities, resources for of the biological system like cell and organism and the ecosystem. Moving to the next one, number 13 is EBI genome. It was founded by Ivan Berne. It provides data and statistics for completed genomes. It was founded in UK in 1992, and besides the information on the ongoing projects, it provides the data and statistics for the complete genome basis. So the European Bioinformatics Institute EBI maintains and distributes EMBL nucleotide sequence database, Europe's primary nucleotide sequence data resource. You can see its URL as well as the logo. Another one at number 14 is genome biology. So this database was founded by Walter Fries in Belgium in 1976. It contains the information about completed genomes, and it also publishes articles describing new databases that have major utility to a broad field of research and have a potential to become the main database of a particular data type. The manuscript here should include the description of development and the testing of the database, as well as a comprehensive demonstration of its utility as well. You can note down its URL being displayed on the screen and just have a look at its logo for the genome biology. Another one, number 15, is ACEDB. ACEDB. It was developed by Richard M. Durbin and Jean-Thierry Meg in 1989. So, although ACDB was initially created as a database specifically for the nematode worm, it has came into mean the database software itself. Now it is being utilized to store the information for other species as well. It actually contains the genome database for Sene hebdis elegans. According to this website, ACEDB provides custom database kernel with a non-standard data model designed with having flexibility in mind. Okay, another one, number 16, is DDBJ. It's a DNA data bank of Japan, which is located in the National Institute of Genetics in Shizuoka, the prefecture of Japan in 1986. It's a biological database which collects DNA sequences, and it is also a member of International Nucleotide Sequence Database Collaboration, or also we call it as INSDC. So this exchange its data with European Molecular Biology Laboratory at European Bioinformatics Institute and with GenBank at National Center for Biotechnology Information on daily basis. So thus these three data banks contain the same data at a particular time. You can note down its URL. The logo is being displayed on your screen right now. DDBJ. It's a DNA data bank of Japan. On number 17, the last one in our list is EXPECIE. It was actually created in August in 1993. Originally, it was called EXPECIE, Expert Protein Analysis System. So, it's a proteomic database. It acted as a proteomic server to analyze protein sequence and structures, and is a 2D page electrophoresis. It's an online bioinformatic resource. It was operated by SIB, that is Swiss Institute of Bioinformatics. It is an extensible and integrative portal. It provides access to over 160 databases and software tools. It supports a range of life science and clinical research areas, from genomics, proteomics, structural biology to evolution and phylogeny, system biology, and medicinal chemistry. So, at the end, I would say data is a precious thing and will last longer than the system themselves. So, on this note, I would like to conclude this particular presentation. I hope you like the content being presented in this particular video. Do click on the like button, subscribe my channel, you can share it on social media, and I must say, and we'll give you a few questions and their answers as well, so that you can practice the questions related to this particular topic. So the very first question is, what is the full form of OMIM? The options that you have is, the options are online Mendelian inheritance in man, online Mendelian information in man, online Mendelian importance in man, and occupied Mendelian inheritance in man. I'll give you a few seconds to think about it, and then we'll reveal the answer of this question. So, the correct answer is online Mendelian inheritance in man. So, the next question for all of you is, which of the following databases were founded by MS Barak? The options are TAIR and Keck, Swiss Prot and SCOP, Swiss Prot and Procite, and PAIR and Procite. So, I'll give you another 4-5 seconds to think about it. If you have carefully listened to, you probably will be able to have the answer in your mind. Yes, you are correct. The answer is Swiss Prot and Procite. Another interesting question, which you probably everyone will be knowing this, is which of the following database contain genetic and molecular data for insect family Drosophila? The options are DDBJ, Genome Biology, EBI Genomes, and FlyBase. And I'm sure if you have properly seen those presentations during the talk when I was telling about these databases, you'll be able to recall the name. Still, I'll give you a few more seconds to think about it. Yes, you're right. The correct answer is FlyBase. Okay, we have another question. Rather, I would say a very simple one. Which of the following is or are the objectives of bioinformatics? The options are organize vast streams of molecular biology data in an efficient manner, develop tools that aid in the analysis of such data, interpret the results accurately and meaningfully, and all of these. Yes, a few seconds for all of you to think about it. Yes, you're right. Its correct answer is all of these. Bioinformatics can not only organize the molecular biology data but develop tools also for analyzing such data and interpret the results as well. And it is being done accurately, and ultimately it has a useful meaning in biological science. Right? So the correct answer is all of these. So at the end, I would like to acknowledge my four students, Divya Jha, Vratika Jain, Akshita Agarwal, and Ruchika Sharma, who have prepared this presentation, and they wanted it to be published here in Pharmacy Lessons Made Easy. So I appreciate all four of you. Thank you. Thank you, everyone. Hope you like this information. Stay connected with Pharmacy Lessons Made Easy for more updates. To subscribe my channel, you'll receive the notifications as soon as I'll upload a new video. Thank you for watching.